You're on the ride with the bloodbath. The Hillsong Express. Oh yeah.
Hello and welcome to the Deep Sea Podcast, a punk take on a science podcast about everything deep sea. I'm Dr. Thomas Lindley and not joined by the Professor Alan Jameson. He is out in the field. He is currently trying to intercept a vessel in Antarctica. As for what's been going on here, lots of prep. We're both in the field in the new year and in some technically challenging places. We'll certainly be telling you more about that as they unfold. But yes, a bit frantic, building lots of gear that I've never built before. I did have a bit of an exciting day when a basking shark arrived in the post at the museum. It was only a tiddler. It was only a baby at a mere 3.7 meters long, but incredibly valuable as a specimen. So working with our colleagues at Auckland Museum, we sort of strategized on the best way to make the opportunity of this beach cast stranding and the amount of things that we can learn from it. We don't often get a specimen of animals like this. So lots of cool things being talked about, tissue samples and what we can unlock from this as a specimen, and also the complexities of keeping this as a specimen. It doesn't fit in a jar, not very well at least. So we're planning its future, but it is here and it is stable in a freezer. And that means we can take our time figuring out what the next steps are. And I got to get my first little bit of work on the museum floor, really, which I found I was more proud than I was ready for, actually. It's only a little thing, but based on that research that me and Alan shared a few episodes ago about the feeding traces on this patch of abyssal seabed that had been upthrusted in Italy, and the interesting feeding marks that have been preserved there, and us guessing if it could be the earliest example of a deep sea fish. So I made a little exhibit for that, and it's really nice to see it on the floor. And thank you to all the co-authors who helped me to get that out there. Part of the reason why this is a slightly disheveled post is because it has been a very busy week. I have been moving house, and so that inspires this month's Song of the Month, which is Sun in an Empty Room, which I find it's like bittersweet. It's like it really sort of sums up that sort of sensation of moving out of a place that was once home, but suddenly it looks all clean and sterile, and you're leaving a lot of those memories behind. So we've been playing that on a Bluetooth speaker while frantically moving from place to place and painting walls white, as is tradition.
With the damage deposit, take this moment to decide.
We want to say a big thank you to our patrons who continue to support us and make the show happen. It would not happen without you. So this month's new patrons are Christian Nielsen and Mol Meir. And check out some of Mol's sci-fi art. It's really cool. They're a molecular planarian biologist, so studying flatworms, and they make really fun art in the form of illustrations, puzzles, and even quilts. So we'll put a link in the show notes to check out their artwork, because we just thought it was super cool. You don't have to go to the full patron level to support the show. We really appreciate even a review or a like on whatever platform you are listening to my voice. It really helps us get more searchable. It gets us at the top of the results. And it's a great way of supporting the show. And the app's already open because you're listening to me. It's right there. Just there'll be something. There'll be something you can prod to say this is good. Even if it's not, just lie. That's okay. It's just between us. On to the deep sea news, and first off I can start with an update. Since talking about it on the last show, there have been multiple apparent attacks on deep sea cables. They are incredibly vulnerable, and so now I guess it's confirmed that hostile nations listen to the Deep Sea Podcast. Slightly daunting that we were talking about it and then it just turned up, but the world's going a little bit interesting right now, so that isn't necessarily a correlation. So 2 cables in the Baltic Sea between Germany and Finland were damaged, and another cable between Lithuania and Sweden's Gotland Island stopped working. German Defense Minister said nobody believes these cables were cut accidentally. The Swedish and Lithuanian defense ministers said they were deeply concerned that the cables connecting the two countries have been cut. And in the discussion on our Patreon's Discord, some of our listeners were showing concern that the deep sea might increasingly become a theatre of war and espionage in the future, and that this might actually be a challenge to the deep sea. And it's probably going to drive lots of technology and research at the same time. Military spending is always big, but it's some place that's often viewed as separate from us. Again, we're bringing our troubles to it and making a mess of things. Interestingly, on the back of all this, China also announced that they have a new cable laying technology which could lay data cables to full ocean depth. So now there is no part of the ocean sort of beyond our ability to lay data cables across them. A new squid just dropped. Our friends at the Squid Squad just released the freshest of new glass squid. So the cranchids are an odd bunch. They include some of the weirder deep sea ones and even the colossal squid. So there's celebrities in the mix as well. The genus Theonius was previously thought to only contain 2 species within the Pacific, but the team were able to show that actually there is 6 and went and described 4 new species. So it's quite a mammoth paper. That adds a lot of new species. So that will also be in the show notes if you want to learn more. So some nice upside-down anglerfish footage from the Smith Ocean Institute, which is currently out off Chile, getting closer and closer to Antarctica. It was a Serratius anglerfish swimming upside down, much like the Gigantactus we reported on a few episodes ago that seem to almost always swim upside down. When you're in a dark, limitless void, what is up and down? What does it even matter? But at least in the Gigantactus, they seem to be very deliberately swimming upside down, and if disturbed, will actually right themselves to the wrong way, if that makes sense. So there must be something to it. There must be some advantage to this. In the paper describing it in Gigantactus, Andrew had some theories about, actually it's about getting the lure out the way so you don't accidentally snap your lure when you go for your prey item, when you've lured it in. But it's clearly deliberate. It's not just up and down don't really matter. There seems to be an advantage to being upside down in some of these species. Deep water corals in unusually warm places. So we've spoken before about how the Red Sea and the Mediterranean are unusual in having a sort of unmixed and hot all the way down water column. And back 10 years ago, I'll talk about it in a while, but when we're working on deep sea corals, so the ones that don't have the zooxanthellae, that don't have the symbiotic algae, that don't need light, cold water corals was a synonym, really. You know, people would say deep water corals or cold water corals, basically meaning the ones out of the sunlight. But there are deep corals in warm waters. So this study showed how sort of corals that build frameworks, that build sort of reefs that are found between 150 and 700 meters in the Red Sea. They did habitat suitability to show how extensive they are likely to be. So it's really interesting, one, that they're living in these conditions. They're able to lay down their skeletons in such difficult conditions. So highly saline, really, really warm all the way down. And their study estimated that there was over 250 square kilometers of seafloor that was identified as a suitable habitat for these reef-building corals. And just to put that into perspective, this is in the Red Sea. This is in somewhere famous for its coral reefs and where people travel from all over the world to dive there. It's at least 35% of the area of those photic zone light-loving reefs that are so popular with scuba divers. So this is a huge area that is suitable for these corals, but we haven't been able to look at all of them, so it's not been ground-truthed thoroughly. They're probably not, or they've been damaged in some of these areas. But actually, it's just a good reminder that they are quite a large, important, and underappreciated area of deep-sea biodiversity, or just general biodiversity within the Red Sea. It sometimes gets overlooked because everyone is so enchanted by those beautiful tropical reefs in the shallows that you forget what's going on down there. So that's actually quite extensive corals growing down there. I had to stop myself saying cold water corals then, because honestly, it used to be synonymous. So I had a little bit of history with cold water or deep sea corals many, many years ago, much younger man. I was part of the Coralfish project, which was an EU 7th framework project that wanted to look at the association between deep water, cold water, reef-forming corals, mainly the Lophelia, which I think has changed its name now, and Madrepora, lattice and reef-forming corals down there, so the stony corals. And the idea was that they are enriching fish communities, basically, by providing a habitat. And this was a study in partnership, actually, with the fishing community, just basically saying that this is an important habitat for the fish. Maybe we should close some zones, and it's in your best interest to support us in that, to ensure that your catches stay high in the wider zones. So it was a really interesting project to be involved in. We were selected because, of course, we didn't want to dredge or sample the corals directly, but we did want to see the bone bell fauna, and that's something that baited landers are really good for. So rather than doing any damage to the coral, we would drop down our baited imaging landers. We'd attract the mobile fish within the view of the camera. And then we compare sort of on coral, off coral. And it wasn't a single sort of result. It seemed to vary species by species. But there was certainly a lot of cryptic species hiding within the coral. There's incredible cold water coral reefs called the Belgica Mounds off island in Europe. And we had our landers down there for a little while, and we'd see all the regular fish, the mora, and some nice eely things coming in, and things we see quite a lot of. And then after hours and hours and hours, and all the fun had died down, there were sort of codlings, little whiskery, catfishy looking fish popped out of the coral reef and started to sort of mill around the lander, and they'd just be totally overlooked by any other survey technique. You know, these are, I think these are about 300 meters down, so it's not something you can get down with dive gear. They'd probably hide away if you had an ROV, and it was just really interesting to see these tempted out. And there's obviously a lot more going on with these coral reefs than we realized. There's a lot of things hidden away. So the project came to an end, and hopefully the results were supportive. The sort of research bed that it created continued to tick over. We now know that certain species of skate and small sharks deliberately place their egg cases in the coral reefs, and actually the shape of the egg cases seem to be evolved to tangle and hold onto these coral lattices. And they're also sort of habitat engineers. They change the habitat where they grow. They change the current flow, and they sort of clog with sediment, and they've got all these associated species. So it is like a building block of a habitat. And yeah, that was an important piece of work for me. It's even immortalized in my tattoo sleeve. It ends with a cold water coral reef based on that work. And it's actually that work that brought me back into academia. When me and Alan met out in the Congo, this was the job that was being advertised, and I thought it was time to get back into academia. And this looked like the way to do it, because I just met this interesting fella and this lander technology, and I liked that you could study deep sea fish without damaging anything, and you're seeing them in their natural environment. And so it seemed incredibly powerful to me. So that was a big pivot point in my career. Even though I'm a fishy person, fish don't exist in a vacuum. They've got to live somewhere. So it was really interesting to be involved in this project, and hopefully a project that had some implications that changed policy and changed views, and was science being applied and actioned upon and having an influence. A few listeners on the Discord were asking for an episode about deep sea corals, and around these parts, there is no one else really to speak to other than Tracey, who is a bit of a local legend. She is a coral expert at NIWA, but came into it through a sort of fisheries and fragile habitat advocate and researcher. So champion of the vulnerable deep sea habitats, fishes and coral alike, and just a really charming person who actually really supports the scientific community here. She has nurtured and mentored the next generation of scientists here as well. So we're very lucky to be joined by Tracey for a chat about deep sea corals. I'm lucky enough to be joined by Tracey, who often goes by Di. I think I've always known you as Di. I know as a coral expert at NIWA, but has a very varied career, and we'll probably get to talking about that a little bit later on. Champion of the vulnerable deep sea habitat, fishes and corals alike, but we're going to have a chat today about deep sea corals. Thanks so much for coming on.
Yes, kia ora, Thom, and great to be here. Great to chat about deep sea corals, actually, because you keep thinking, oh, everyone knows about deep sea corals now. We've been looking at them since 2008 in this region, and actually they don't. They're just familiar with the Great Barrier Reef corals. So thanks for the chance.
Yeah, they have a sort of forgotten status, even though we've been going on about them for so long. I wanted to start with a bit of a foundation, because a lot of people are sort of aware of corals, but not their structure as an animal. So can you talk through just what a coral is, how this colonial organism sort of functions?
That's right. They are animals, and the little polyp that lives on the animal is a bit like an anemone. So each polyp is an individual animal, but they have an exoskeleton. So they build in a tree-like way, often, a skeleton with branches. And it's on those branches that the polyp lives, often embedded in the skeleton, and so you can't see it, but then it protrudes with the tentacles when it's feeding and breathing. Yeah, the animals like anemones and jellyfish are in the same group of Cnidaria. And their structure is, the skeletons are formed often using calcium carbonate, which they get from diet, but also from just the natural minerals in the ocean.
Is there a sort of continuous tissue? Are the polyps connected to each other?
In some, yes, there is. The senenchyme, or some people call it the cenosac. That's the tissue that is covering the skeleton and connecting the polyps as well. There's not loads known about how that connection exists, but if the senenchyme disappears, the coral's not doing very well as an animal. So yeah, within each polyp, there's this pink tissue, for example, on some corals that connects along the branches to the next polyp. That's external. Internal, there's also stuff happening as well.
Is it one sort of cloned organism? Is a single colony sort of genetically uniform?
Yes, we think so. That's correct, yeah. And there are male and female colonies also.
Oh, right, at the colony level rather than the polyp level. Oh, that's interesting.
Yes, that's what we believe, certainly for the scleractinian stony coral. So one clump will be a male clump and the other one will be female. So the female will produce the oocytes and then the spermatophores come from the adjacent clump. Yeah, but that kind of connection is quite tricky.
Yeah, I bet.
And then some brood the animal inside the mouth of the polyp. So you've got that going on as well.
They're quite tricky in their simplicity. So there's just one entrance / exit. So they eat and they expel eggs and sperm and, by the sounds of it, expel even juveniles just through this single opening in the middle.
That's right, yeah, the cavity. The ones that brood the larvae, that would be in what we call the mouth of the polyp. So that's just sitting in there amongst all the rest of the organs of the coral. And then, yeah, it just pops them out when they're ready.
Oh.
And off they swim, which is amazing.
Yeah, and they must have to halt all digestion. They must have to halt a lot of other processes in order to do that.
Focus on the breeding. Yeah, that's right.
Yeah, single cavity. Is a single colony functionally immortal? Or because it's a clone, is it sort of building up genetic damage? Is it important to have that sexual reproduction as well to get some variety in there?
You would have to have that. You also would have to have the animals being far enough apart so that the genetic material is more variable.
Yeah.
I'm just going to butt in here and say I'm not a taxonomist and I'm not a geneticist. I'm not a molecular person. My career began with us not even knowing about these animals. We didn't know where they were. We didn't know what they were.
Started on day one.
Yeah, it was, the focus was on single species stock assessment when my career began. So we were looking at how many fish are in the ocean, how many orange ruffy are on these seamounts or in this area, and then focusing more on the bycatch, what we were also getting in the trawl, the other fishes, as well as the invertebrates, led us to do a lot more work on invertebrate species in our region. And that's when we started with the coral research, the spatial distribution knowledge. Where are the hotspots?
This came from a sort of fisheries element because it's important for the fish. It's important habitat. These are not just animals in their own right. They shape the habitat around them.
Exactly. Yeah. So, they are very important. You'll hear that word importance of habitat throughout. Anyone who talks about corals will mention habitat and importance and associates what lives on the corals, what species use them for shelter and refuge, or what species really rely on them as well, for platforms to get into the water column to feed. So all that kind of information and knowledge has grown for me since we did the fisheries work. Of course, there were taxonomists in the museums in New Zealand looking at the corals around our coast, and that work was going on. And when they put the cable between the North and South Island and lifted it for maintenance, they found corals on that cable. So there was work going on in the early days, in the 60s and 70s, but that was more coastal. The deep sea and that knowledge really just started with the growth of the deep sea fisheries and knowledge of what was on the seamounts.
Yeah, things were just coming up in the bycatch that never seen before. And
exactly.
Some of the images from those early days, huge corals coming up with the trawls.
And those huge corals still come up with the trawls. Because, people are always going to new areas to look for more fisheries or more stocks, and they're bringing up corals still. We're continuously doing work on protected corals for the Department of Conservation. We're monitoring bycatch on fishing vessels, and we're still seeing those really large corals coming up from the deep. For example, the bubblegum coral, that charismatic paragorgia, that's still being brought up in trawls as we speak.
And what would the age of a colony like that be? I know they're incredibly slow growing.
Well, the bubblegum corals don't seem to be as slow growing as some of the other coral groups. So we'll be getting black corals living to over 2,000 years. But the bubblegums seem to live, to be honest, we've only radiocarbon dated one specimen, 300 to 500 years. So yes, old, but not 2,000 years. They seem to have quite a fast growth rate. It might be because they're not calcium carbonate structures. They're more gorgonin, chitinous.
If they're well fed, they can lay it down pretty quick.
Yeah.
Should we talk about the two main structural groups? How they make their skeletons sort of impacts the flavor of the coral quite a lot. If they're slow growing and hard or sort of proteinaceous and a bit flexible.
Right. So there's the scleractinian stony corals, that protected reef-like coral that we have in our region that's made of calcium carbonate. And so they are very slow growing. It could take about 2,000 years for 1 meter of that colony to grow. So that's one key group, the calcium carbonate structures, scleractinian stony corals. Then there's the bamboo corals, which are a mixture of calcium carbonate and gorgonin. And they are also very slow growing, but not as slow as the stony corals. And then we look at the black corals that's made up of a different material, kind of more chitinous. They can be quite fast growing in the Fiordland region and live to about 300 years. But in the deep sea, they're very slow growing, and we've aged them to over 2,000 years as well.
Even the very youngest of the ones you mentioned are older than our ability to impact them. We've only been able to bump into these things for the last 100 or so years at the very most. The Challenger, maybe it was a few pretty specialist expeditions, and it's only really now last 30 years, maybe last 40 if you're pushing it, where we've really been impacting these very, very slow growing animals. So they've not really had a chance to rally or adjust.
No, not at all. We've carried out recovery experiments. We've been out to the seamounts and looked at where there's been damage to the colonies, and then we've looked for signs of recovery. Are the corals growing back along those tracks that have been heavily trawled? And we're not really seeing any signs of the growth. What seems to happen with the stony corals, which my colleague has been studying, Jennifer Beaumont, the reproduction work, was that we've seen the larvae brooding inside one of the species, Goniocorella, and we've seen those larvae come out and settle really quickly and start laying down a skeleton to get some kind of base, get some hold on something hard and on a suitable substrate. And then the growth would slow. So it's initially, let's get settled and start growing a skeleton so that we're safe and we can start growing. That seems to be a reasonably fast phase of growth, that initial one, and you see that in a lot of animals, but then it slows.
Let's talk about the corals as habitat engineers. So say an empty seamount or a seamount that's been impacted by industry. What changes are coral colonization sort of offering? How do they change the habitat?
So a lot of the seamounts we've studied on the Chatham Rise, a few are protected. And so we can look at those pristine environments and see how the corals are behaving there and see what they provide as far as habitat for many other invertebrates, not just the live matrix, but the dead matrix as well. So we can observe side by side an impacted seamount, what importance the corals have. They don't just provide habitat for other invertebrates, they're also important for other fisheries. The fish either feed on the animals that feed on the corals or feed on the animals within the corals, or they use that coral matrix. They're using that for refuge and shelter as well. So quite an important role.
It's beyond the immediate habitat. They engineer and they influence quite a halo around themselves because they change fluid dynamics as well, and they bring more nutrients into the area really by changing the fluid flow and the productivity in the area. So they seem to enrich quite far beyond their colonies.
That's been hugely studied in the Great Barrier Reef area, where you're seeing a coral and the coral dinoflagellate symbiotic animals. You're seeing them maintaining at an individual level that kind of environment. We really don't know how much that is happening in the deep sea. That's kind of a huge area, but you're right. Those corals, that halo of corals that are draped over the flanks of the seamounts are also changing the whole environment at a local level. Upwellings that occur with the oceanography around seamounts is a huge thing, and that's where you'll get a lot more feed, but that's beneficial to the corals and the animals that live within the corals.
I realise we've missed something quite fundamental when we're talking about deep corals versus shallower, which is the lack of the symbiotic alga.
That's right. All of the corals that we look at in the deep sea, they are azooxanthellate. They don't have these dinoflagellates, so they do their own breathing and feeding. Whereas in the shallows and Great Barrier Reef, they're helping the corals survive by doing the breathing and providing the food, and also maintaining the ocean environment around the corals, so keeping that pH at a level that the corals like. But they're battling climate change at the same time, so that's when you get the bleaching events, and those dinoflagellates die. They're the ones that bring the colour to the coral, and so you just see bleaching and pale corals. So we're not seeing that in New Zealand because our corals are doing their own breathing and maintaining their environment. But they still get stressed because the ocean chemistry is changing as well as the temperature in our oceans. And so when that changes, you can't form the skeletons. So they are suffering in a different way, but in just as serious as the shallow corals.
Maybe less visually striking. The bleaching events are quite immediate and quite sort of sudden, but is it more subtle with the deep ones? It's hard to see when they're stressed.
It is. Our deep ones are very colourful when they're healthy, actually. They're very pink and red and yellow and purple. They're pretty stunning. So you would see changes because of temperature, and that senenchyme that we talked about earlier, the tissue that surrounds it, that pink tissue would disappear, but be more subtle. We're seeing those massive bleaching events are very dramatic, seen from space, and
and like happening over a week.
That's right. Yeah,
it's going to be more gradual, and it's hard to right the ship in the deep sea. And we're seeing more and more. Well, maybe it's always been there, and we just haven't recorded it. But we're seeing more and more sort of high temperatures rolling through the deep sea, and because of the crazy current regimes where it's often detached from the surface could be fine, but a pulse of warm water has gone through the deep sea and stressed a load of things in a way they haven't done before.
That's right. We're seeing those hotspots in New Zealand already. So it is serious for the shallow and the deep sea corals.
When you're growing over hundreds of years, they can't dodge out the way like fish finding a new optimum.
Well, I mean, you mentioned earlier in the interview this transplanting at sea that's going on, and it's not just going on in shallow water corals, but in the deep sea as well. They're taking up colonies and growing them in an aquarium and then settling them back into those deep sea environments.
I did not know that. I know you in NIWA, you've kept them alive in the labs there.
Yeah, we maintained them in our aquarium, and they lived for well over a year in a cold, dark environment that mimicked their natural environment. And the first work we did was on ocean acidification impact, so looking at changes in pH and how they would affect the corals. So that was one experiment we did, and over a long period we certainly saw impacts, dead polyps and the senenchyme, that pink tissue, disappearing from the skeletons. And then the second study was looking at sedimentation. So how did elevated levels of sedimentation impact the corals? And again, we saw, pretty robust life with the coral results. The corals were doing okay for a short period of time. But as the sedimentation accumulated over time, we could see stress on the corals again. The tentacles weren't able to flick out the sediment adequately and they would die, or that senenchyme tissue would just be disappearing over time.
They can cope to a point, but then it's quite an abrupt point where it's just too much and the colony is struggling.
I mean, they often live in quite a high sediment environment. Some corals live on seamounts where it's hard bottom rocks and not much sand or sediment. And then other corals will live on small stony structures on flat sediment. So to a certain extent, they can maintain themselves and stay alive in those environments. But when you add additional stress, that's when they struggle. So they're not all found on hard rocky outcrops or on the sides of canyons. They're also found in soft sediment, We have the stony corals, the cup form, not the reef form I've talked about a lot. Those cup corals can be on hard structures like other corals or on rocks. You have the reef-forming corals that attach to rocky substrate and seamounts and drape themselves all the way down the flanks of seamounts. But you also have the cup corals, and they can live on hard substrate or just in sediment. They have a different structure.
Are they the ones that tend to be solitary? They look like an armored sea anemone. They make these cool tooth-like plugs.
Yeah, solitary cup corals, yeah.
I know it's present in a lot of the stony corals, but because they're so much bigger, you really get to see how beautiful that structure is. Like how much the flesh of the animal is quite small, and there's this beautiful sort of ridges to the cup itself.
Yeah, the septa.
Is that what it's called?
Yeah, and they have these different patterns. Yeah, they're amazing. And then the column can be long and elongate or flat and like a bowl. And yeah, they're really beautiful. You're right.
If I'm waiting for someone at NIWA reception, I always drift over to that display case.
Yes.
There was something else you curated at Te Papa, wasn't there? There was another display.
Yes, the invertebrate display we helped curate. Corrine, Sadie Mills and I prepared some of the display cases for the current exhibit at Te Papa in the biodiversity area. And Weta Studios helped create some of those corals, the soft ones. For example, the Corellia morpharian coral, that's the one that didn't form its skeleton again and stayed anemone-like after the last ocean acidification event millennia ago.
Wow, gave up.
Yeah, it probably said, I quite like it without a skeleton.
Yeah, I don't mind being floppy.
So the Corellia morpharian is beautiful, and Weta Studios created a 3D model of that and a couple of other animals, soft animals as well. Well, I love the Corellia morpharian because it is related to the scleractinian stony corals that we've just been talking about. It's the kind of link. So it looks like an anemone, but it's a coral.
And there's always those exceptions. It's
exactly
the stony coral that isn't stony.
Yeah. And of course, a black coral will form a tree-like structure, but also be uniplanar or pen-like, linear, like a sea pen. It doesn't branch at all. And you get those forms in the coral world, and you think, why do they develop like that? What's the advantage? And
when it isn't a single coordinated organism, they can only grow, they can only move by growing new polyps. And what makes that polyp decide I'm going to be branchy or I'm going to be whip-like?
Exactly right. So there's many different genera, and they're quite different. And I suppose the whip-like one can flop over in the currents and then bounce back and might not be impacted so much by trawl gear because it can pop up again. But, it wouldn't have developed because of trawling, but it might have developed because of currents and in the ocean and the deep, where it's strong and struggling to stand up.
It can't have too much drag if there's regular sort of flushing events with high turbidity. Yeah, that might be one.
But then there's others that form a trunk, like a tree, a big base, and then start growing up. So they focus on getting stability in the deep, setting themselves in a strong kind of position.
Which might not be advantageous because it's so strange where they all start as this individual polyp.
Yeah.
And that's advantageous for the sort of end result, for the end colony. But for that individual, it's probably not going to see that. It's probably going to be replaced by multiple generations before it sort of reaps the benefits. So they're almost putting the work in to benefit their great-great-great-great-grandchildren.
No, that's true. Although sometimes they can grow fast at one stage in their lives and then slow, or they put energy into growing out on one branch and not so much on the other. Yeah, so some parts of it, they might have to not wait to the grandchildren. I'd say most of it's all about food. Can I get up high enough in the water column to get the food?
Yeah, is this side doing better than that side so it grows quicker?
That's right, yeah.
Is there any sharing of resources through the connecting membrane, or is each polyp on its own?
Yeah, we talked about that a little bit earlier. I think there's sharing of resources, but I have no real understanding of it. They seem to be able to survive on their own when we've taken the samples back, and, they haven't seemed to be dependent on the polyp next door. But there must be connections. There's often hollow tubes between some of the corals, not all, but that stony branching form, the branches are hollow.
Connection was sort of running through them. I always thought of like the nerves of a tooth. There's a little core of flesh that goes through.
But the other corals, the bubblegum and the black corals, that skeleton's solid, and you can't really see that they would share. It's a hard structure. So perhaps it's the external tissue that shares between polyps and not the internal.
Oh, that's interesting.
Yeah, it's tricky. I mean, if you cut open a Scleractinian stony coral branch, you'll see those tubes, but you'll also see divisions, like it stops. It's like a wall. And so you think, what's going on there?
What are the sort of common commensal organisms? Who are the friends of the corals? And I suppose there's going to be predators and things that graze on them as well.
Yes, the echinoids graze on them, so I'm not a fan of them. So those deep sea urchins, they'll move across the seamount and start nibbling on the coral polyp. The worms that we've already mentioned about, the polychaetes, the marine polychaetes, they seem to be more commensal and it seems to be okay. The galatheids, the little squat lobsters, use them as a platform, but they're not really, in a symbiotic or commensal relationship. They just use it as a structure to get into the water column to feed. Lots of ophiuroids are found, the little sea stars are found on the corals. That's probably it for the kind of the commensal animals. If you're looking at those Gorgonian octocorals I mentioned earlier, they seem to have these ophiuroids that are always on them, and they just seem to live in more of a commensal way. They're not being eaten. They're just, those snake stars are just twisted all around the branches, and
I've seen
really that. Abundant. Yeah, so that's on those, what used to be called Plexauridae. They've changed the family name now. So they have them. And black corals in the Fiordland region, it's been observed that they have that same snake star that will live in association with those huge black corals forever. They've been studied by scientists who have been in the Fiordland area since the 70s, and they see that same colony with those same snake stars. They're all happily kind of living together.
Oh, it's a long-term one. And then I know a lot of fish, particularly the elasmos, there's some skates and some small dogfish that seem to really target the coral lattice to lay their eggs. And the egg cases actually seem to be structured so that they coil and wrap around the coral and get secured that way.
That's exactly right. In fact, my colleague Britt Panucci, whom you've interviewed, I believe, always says, look out for the skate cases on the coral matrix. Yep, that's right. So very important structures. There's always a seamount we go to on the Chatham Rise, and we see these two giant lepidians, these huge fish. They're always in the shot, and you think, what are they doing there? Are they just sheltering and using it for refuge and passing through? But they're large and they always seem to be hanging out there. So they must love that environment. They must be feeding on something there. But smaller fish, we've seen little morid cods sheltering in their branches.
Yeah, cusk eels too. Cusk eels seem to have a home coral sometimes.
That's right, yeah.
Did you want to talk a little bit about sort of career progression? Because you were on the front line of getting to sea, and I know you've done a lot of work for improving the offshore environment, particularly for women in science. That was a listener request. You've seen the industry change, and you've sort of driven a change in that area. Because even the 20 years I've been going to sea, the attitude has really changed. We still get fisheries observers writing in saying, like, I keep finding garlic under my pillow because they think I'm a witch. And
yeah, well, that's pretty sad, isn't it? You think things have changed. I started in the 70s, so it was a long time ago. And so I was 4 years working in the Fisheries Research Division. It was called the Marine Department, and then it became Ministry of Agriculture and Fisheries. And women had just started to go to sea vessel, the James Cook. I think the first woman, Anne Brunt, who went on that vessel, the director had signed off their voyage program without reading the names. And that's how we got a woman onto that trip. And then for those 4 years that I was working, there were always 2 or 3 women going on that research boat. And that was a pretty good, equitable experience, to be honest. Young technician that I was, learning about the job and learning how it was to be on research vessels. It was all a good experience. You often come to awareness about equity when you're a little bit older, and you go, hold on a minute. They're they're doing the same job as me. And I've been doing this for years and they're paid more than me. So that kind of awareness came when I got back from overseas, when the exclusive economic zone had just been declared. The EEZ suddenly was 200 miles of waters to research.
Well, so you were part of that transition, because it's so galvanized now, the EEZ. But it's not, it's a really new thing. Like there was a time when it was the 12 mile.
We didn't have a research vessel that could really perform in that whole region. You know, it was huge. And so we relied on initially foreign vessels that were in the region, on Japanese vessels. We were doing research, Russian, German, the Wesermunder was there for 2 years assessing the size of our resources. But yeah, I was often the only woman on those trips, and kind of relied on the support of my male colleagues to make sure everything went okay. Yeah, because it was very unusual for women to go on those voyages. And then we spent 10 years going on the New Zealand research commercial vessels before we got our research vessel, the Tangaroa. So we used commercial boats to do research for quota or research for money surveys on all the new stocks that were being discovered, orange ruffy, Oreo species. This was mainly a deep sea focus. If I was leading the voyage, I always made sure I had women on my team. You know, we're part of 50% of the population, and of course we bring value to the job, and we do things slightly differently sometimes and add a huge dimension. And sometimes that was hard to get the woman on the trips, and sometimes it was a struggle in those environments because you did have sexism and pornography in the workplace.
Yeah. Even during my career, that's really changed. Like there's just the atmosphere offshore has changed.
Yes. Yeah. Well, good. So I think, that was a time of making sure people heard women, and we showed that we were capable of leading voyages, and several women that I worked with led voyages in the 70s and 80s. One of those women would say maybe it was a bit easier then. We had equal employment opportunities programs active, and so there was a big focus on equity and fairness and women's rights. And then, of course, so that was a bit of a stick, so the men had to comply with what the government was saying. EEO programs were strong. They had a lot of clout. But then over time, sometimes it was hard for women who had had families to keep going to sea. So I didn't have children. And so often, even in the 90s, I was still often the only woman at sea. But yeah, it was, as you say, the environment was loads better. We'd kind of addressed the issues of no pornography in the workplace. We had rules, and sexist behavior wasn't as common. And now we seem to have that, a lot of women are going to sea and they're leading voyages, and they sometimes have an all-woman team. But that still gets questioned, which I find really intriguing. Gosh, how would it be on that voyage? It's all women. Who would do the lifting, for example?
What if you get a really big rock?
So we still get that, that bias, and it's a bit more underground, you could say, that unconscious bias. But yeah, throughout my whole career, I've tried to support women, because it has been a very non-traditional role. And, 2 women at sea was much easier than 1, or you'd quickly get to know the steward, who might be a woman, and you knew that you had allies on board. So that really helped if there were any issues or any kind of harassment issues. And then as I pass on the mantle of the coral research, I'm probably a bit biased myself, and made sure that I've passed it on to a lot of, great women researchers. So there's women who are coming through now who can pick up most of this work. Here's a shout out to women researchers. Thanks for the question.
Well, yeah, I want to talk about that, because in my time, I've seen a huge change, and I just think the more different your team is, the better you work. You can have the smartest, most capable people, but if they're all the same type of smart and capable, they only have the same idea. Yeah. I really like sort of mixed, diverse teams, because someone at the back of the room will have this totally different experience with a different way of thinking.
Exactly.
Thanks so much for your time today.
Thanks, Thom. Nō reira, tēnā tātou katoa.
Oh, nice. I'm still brushing up my te rea. I need to get more confident with it, even if I'm doing it wrong. I just need to go for it.
Yeah, get your greetings sorted. I presented this really nice one at my retirement party. when the old net is cast aside, the new net goes fishing. And that's kind of about the old net has one way to do things, but the new technology can do it slightly differently. And that's how I see passing on my work to these great mid-career women who add this great new value. So it is kapu te ruha, tahau te rangatahi. When the old net is cast aside, the new net goes fishing. Kia ora.
No episode would be complete without grabbing a quick coffee with Andrew and seeing what he's been up to.
Coffee's ready.
Since we last spoke,
Yes.
you've had a grand adventure.
Have I what? Yeah, 36 days out at sea on Tangaroa, working with a fantastic team of people, looking for where freshwater eels go to spawn. Now, the traditional wisdom is they do it up near Tonga, but tags have shown that no, this is not the case. They're heading more up towards New Cal. But my involvement in that voyage wasn't so much with eels and their larvae, but the bycatch, which was super exciting, actually.
We'll set the scene a little bit, because I think some people in the UK, it's the same Anguilla genus, so it's the same as our freshwater eels in the UK. And they've got a wacky life cycle where they get really big and chonky in fresh waters. Their bodies start changing. They change colour. Their eyes get bigger. They stop feeding. All their bones reduce, and they just put everything into reproduction. And then they leave out to sea. And we don't know where they go. So we only go to the Sargassum Sea for the ones up north in the Northern Hemisphere. So that's why it's deep sea. That's why we're talking about a freshwater species, but going deep sea.
Yes.
And then there's a missing bit. And we know they have these crazy larvae, the
They're just bizarre. And this is what unites all true eels, is this larval form, Leptocephalus. Looks like a willow leaf made of gelatine. You can see through it. You can see the myomeres. And then at one end, you've got a great big eye, relatively, and this weird little mouth filled with oversized teeth. And these little beakers just swim and eat and swim and eat and swim and eat. And then at some point, when they get close to land, they undergo metamorphosis into a glass eel, where they suddenly go from this bizarre willow leaf to literally a little transparent eel.
And that's when they come back into fresh water, and that's when a lot of cultures eat them. So in the UK, that's your jellied eels and things like that.
Yeah. Don't think they were harvested at that point by Maori. They are still a Tonga species, a treasure. And the New Zealand has Anguilla diefenbachii, which is the biggest and heaviest freshwater eel in the world.
They are chonkers. And they're tame. You can feed them.
I know.
I've fed it a sausage.
They used to be slaughtered as a pest because they were eating these beloved trout, these pest species that we introduced. But there's been a seismic change in public attitude to eels now. And
They're just so charming. They're very, very funny when they come up and they'll take a sausage from your hand.
Fingers, yeah. and you can stroke them.
So this was a big marsden project. You sort of joined as part of a much larger team.
Yeah.
And it was trying to solve this mystery, looking for this Leptocephalus larva. But that is exciting from a deep sea context, because where do you go and look for that larva?
In the deep sea, relative deep sea.
Yeah. And when the deep sea comes up to you. So you were shooting the gear at night. You were waiting for the deep sea to come to you.
Yeah, that scattering layer coming up. So we were shooting that gear down to occasionally to 1,000 metres, but mostly 500, and then processing it. So we got about 190 mesopelagics, true mesopelagics, as opposed to all the larval stuff. We were getting a lot of larval, tropical reef larvae.
Yeah, reef fish.
Reef fish, ballistids, lutejanids, ceranids, tiny little fistularia about that big.
But you've got the classics as well. You've got dragonfish,
Yes.
you've got loads of minktophids.
Yes, eventually. Yeah, got
So that's the lanternfish.
hatchetfish.
Oh, yes, nice hatchetfish.
Nice
Might be
hatchetfish,
new.
which might be new. Now, this is going to be quite exciting. These were all coming up wrapped in jellies. They were wrapped in bubble wrap, nature's bubble wrap. So they were in perfect condition, and yet I was getting a swag of them with only one spine. So my question was, why that spine? Have I got something completely new? So the really cool thing is I got a culeatus and I got this thing in the same toe multiple times. So we'll be able to run tissues to see if CO1 shows whether or not they're different. If they're different, this will be the first new species of Agarapelagus to be described in nearly 100 years. Nice. The last one was Norman in 1930, Agarapelagus gigas. So I did a little dig around for a bit of skite.
So we should dust off this genus and get some
I
newbies.
Yeah, I think it's about time.
Yeah.
We've got plenty of Polyhypnus, And synoptics, well, there's only the 4 of them. Come on.
New hatchet just dropped.
New hatchet just dropped, yes.
Nice.
The other thing we got was some dragonfish in the genus Eustomice, which, because of the temperatures in which we were working, which was 35 to 38 degrees, we were under the pump quite considerably to get this stuff identified as best we could and then frozen. So we had chili bins that had a bit of water in that had been frozen, trays with ice. Worked really well, but once you took them out of that and started looking under the microscope, you were in that oven.
Yeah.
Our lab was called the sauna lab because it was just so hot.
You were down in the fish factory.
Yeah.
It was blazing heat.
Blazing heat. I think my longest stint was 30 hours with 2 4-hour blocks of sleep in there somewhere, as I recall. I seem to recall collapsing into my bunk after a cold shower and then waking up with a start and feeling guilty and rushing back down again and getting back into it. But yeah, it was exhilarating and exhausting working with a number of young people who are doing PhDs. They are just the most wonderful people. They really were. They were fun to be with. They were energizing because of their enthusiasm. It was just great. I didn't feel like the old guy of the trip anymore. But yeah, it was just great fun. Really exciting. So for the squid squad, we got a number of good squids. We got a Histiotuthus, one of those cockeyed squids.
And a big one.
It was huge. And that was a good 30 centimetres tip to tip. That was really big.
I'll be excited for that.
We got a number of cookie cutter sharks, quite small ones. I did return 2 of them alive, but I kept 3 because they were rather small.
And they cookie cut the net.
They cookie cut the net. I found an interesting cookie cut in amongst all the bycatch in the bucket. And I went up on deck and saw the bosun and said, Glenbo, you're looking for a hole that looks like this. And he comes back down after about 10 minutes going, found the hole, Andrew, come and have a look. So I've got a photograph of him holding the piece right by the net, and it fits like a jigsaw puzzle.
Absolutely, they don't waste.
And a big shout out to the crew of the Tangaroa. They fixed that IKMT a number of times after it tore. So did some of the young ones too. They were very dandy with the needle and thread.
Oh, cool. You made a really nice observation on one of the dragonfish. Do you want to talk about the lure?
We were getting a large number of Ichthyostoma barbatum. This is a dragonfish that's got a very distinctive diagnostic lure, quite short. And as you come down the stem of the barbel, you get a purpley blue chunk and then a sort of ornamental reddy orange chunk. And I was picking through the catch, looking at it going, okay, that's another Ichthyostoma. And then attached to part of the Ichthyostoma was a little copepod with eggs. And the eggs were bright blue and the copepod was brick red. And I put the 2 together.
It's the same toe.
And it's exactly the same overall morphology. This dragonfish is mimicking a copepod with eggs. And so it's flicking it out there. So the prey comes along thinking, I'm going to have a bit of copepod snack. Thonk,
as good as a copepod gets, loaded with eggs, loaded with energy.
Oh, yeah, they're protein plus.
Yeah.
And then it's suddenly...
Right down to the colour.
Right down to the colour.
The colour is correct.
The colour was correct. And that was so cool to actually find that and see it. Because you hear that these things...
It's been hypothesised, hasn't it? But seeing them next to each other was wild.
Yeah, and that was really cool. Had a little bit of excitement on one toe because we get this bucket and it was just jellies, salps, ctenophores, Venus girdle, all that kind of gelatinous organism. And you're scooping through it trying to pick out the big fish to get them out of the way so Neve and I could start processing that. And then the lepto team would just ladle it out and look for, in smaller quantities, looking for the leptocephalus. So I'm scooping through this and I'm get these really turgid strap things wrapping around my hands. And I'm going, what's that? It's not a leptocephalus. And then I saw the bell and I went, oh. Picked it up by the bell. It's a box jellyfish. Fortunately, it fired off all its stingers in the net.
It had been rolled around so much that everything had fired off.
Yeah. So I pulled it out and I put it in a bucket of water and it's still alive. Oh. And you're talking about tentacles that are, when they were untangled, would be well over 3 meters long. So, that was a good wake-up call, that one.
And that would have been a day spoiler.
That would have been a trip spoiler, potential life spoiler. Photographs I've seen of people who've been stung by them, they look as though they've had electrical wires.
Yeah, it looks like barbed wire almost, like permanent tendrils.
Branding, these deep red scars that are lifelong, and the pain level apparently is next level. So, that was a good call. I wore gloves after that. Yeah. The other close call we had, obviously, was the cookie cutters. They were still thrashing around, and one of the team wanted to release them and put their hand in the bucket. I had to go, no! And then it stuck its tail out, and they put their hand out. No, you never, ever, ever grab a shark by the tail.
Particularly something dogfish-shaped.
Well, that particular shark and that whole family, and I described it, I said, the lower jaw is like an ice cream scoop with piranha's teeth around the edge. And it will happen so fast, you won't even sort of register. It's just removed a scoop. You don't need stitches, you don't need a plaster, you need plastic surgery after that.
Yeah, because there's nothing to stitch together, it's gone.
It's just, this ice cream scoop's come in and removed a chunk. You do have to be a little cautious. Yes, the other curiosity I found was the dragonfishes, all of them, without fail, wrap their damn barbel round in their mouth on the right-hand side. So trying to unpick and gently tease out this barbel with the diagnostic characters on it and lay it out and then flip it over and try to,
So fragile, and there's no hope of IDing it if that's...
Once that's gone, you are whistling Dixie if you want to
They
try
all
and
look
ID
the
it.
same. The bit with the name on fell off.
Yeah, pretty much. The name tag. Hello, my name is.
Yeah.
That was a great trip. That was really fabulous. And I guess now the hard work starts.
It does, but not before you've had your little bit of time to catch up. I was with you for your first beer.
Yes.
And it was a pleasure to
nectar.
watch.
Nectar.
It was a pleasure to watch you have your first beer. Well, welcome home.
Thank you, mate. It's great to be home. If you get the chance to go to sea, grab it with both hands.
And that concludes this episode of the Deep Sea Podcast. Just a little reminder that this isn't just a podcast, it is an ecosystem. There's all sorts going on. So just to make you aware that there is a website, there's extensive notes to each one of these interviews. There's wider reading you can follow. There's also stuff that goes onto social media. There's video content on YouTube, and there's even a newsletter which will keep you up to date on all of those things. So if you dive into the show notes, you'll see some links that can lead you to where other stuff is going on. So we're on Twitter, we're on Instagram, we're now on Blue Sky and YouTube as well, and there's lots of extra content on there. But until you come and harass me on social media, and please do, I'm fairly friendly, we'll deep see you next time, and I miss you already. If you would like to advertise with the Deep Sea Podcast, feel free to get in touch. Our audience is primarily young people with an interest in science, often undergraduates or people considering a degree in marine science, but it also includes established scientists. Feel free to get in touch if you're interested in reaching these groups.
Hello, my name is Theo, and I was having a sleepover with my friend when my mom turned on white noise on Spotify. 8 hours later, the white noise turned off, and I was half awake and half asleep, and your podcast started playing instead of white noise. So I was having these weird dreams where you and the other doctors were giving me information, and it was feeding into my dream about first-person shooters with squid everywhere. And so I woke up, and I listened to your podcast for the rest of the night.